Hello, my name is Anna Horvath, and this is my microbial disease presentation. The diseases I will be covering are diphtheria, whooping cough, and croup. And in the various images on this slide, you can see each of the microbes that are responsible for the various diseases. So the first one I'm going to be talking about is diphtheria, which is caused by Corynebacterium diphtheriae. This bacteria is a gram-positive bacillus, as you can see in the image on the top. It is also club-shaped, so that's why it's not doesn't look like a traditional bacillus. This bacteria is non-motile, and it produces an exotoxin called diphtheria toxin. And this toxin acts as a prophage, or that's its mechanism. And what this means is it inserts into the host cell genome and it inhibits RNA translation, so the host cell cannot produce proteins. So the disease mechanisms, epidemiology, and symptoms of diphtheria. This bacteria infects the nasopharynx or the skin, and it is spread via respiratory droplets or from contact of an infected person's wounds. This toxin causes lesions in the infected areas and is only transmissible between humans. As you can see in the images, the top one shows lesions that would be in the nasopharynx area at the back of the throat, and the drawing type image on the bottom would be a skin lesion, which is also caused by this bacteria. Symptoms of this disease include a sore throat or pharyngitis, which is an infection of the pharynx, a fever, swelling of the infected area, so the throat or the skin, and then if this bacteria gets into the bloodstream, it actually can cause heart problems such as a racing heart. So to diagnose diphtheria, the bacteria must first be isolated and then tested for toxicity. This is done in the image on the top there. You can see a filter paper is put on the culture medium, so when it's isolated, and then it is streaked at an angle to the filter paper. Then the toxin can be detected because the filter paper is soaked with an antitoxin. So it forms a precipitate with the toxin on the filter paper. So the toxic, if it is toxic bacteria, then it will have a precipitate. And then the ways that it can be isolated, there are 3 different types of agars that are usually used. There's a Loeffler agar, which is nutrient, Mueller tellurite agar, or Tinsdale tellurite agar, which are both selective. And then the disease can also be detected finally via an ELIC immunodiffusion test. So the treatment for this disease is usually done with antibiotics. These include erythromycin, which inhibits protein synthesis, which would probably be the toxin in this case, or penicillin, which inhibits cell wall synthesis, as it is a gram-positive bacteria, so penicillin would be effective. And another way to treat this disease would be an antitoxin, which, as we saw on the previous slide, reacts with the toxin, and it actually neutralizes it. And this would be injected via the bloodstream, so into a vein or into a muscle. And finally, to prevent an infection with diphtheria, there is a vaccine called DTaP, which is for diphtheria, tetanus, and pertussis, and it is an inactivated toxin vaccine. So the next microbe I'm going to be covering is Bordetella pertussis, which causes the whooping cough. This is a gram-negative Coccobacillus, as you can see in the top image there. It is an aerobic microbe and is also motile. So in that bottom image, it's kind of a fluorescent picture of Bordetella pertussis, and you can see that there are some flagella coming off the end. The toxin that this microbe produces is called pertussis toxin, and the category of toxin that this is in is ADP ribosylating. And what this does is it affects signaling, so signal transduction between important cellular proteins, and basically completely inhibits communication in the cell, or important communication. And then this bacteria also produces hemolysin, which affects red blood cells. So disease mechanisms, epidemiology, and symptoms for whooping cough. As you can see in the image to the right, it is spread via respiratory droplets, such as sneezing, coughing, or spit. It is most contagious when somebody is first infected, and the individual would most likely have very mild symptoms and not even know maybe that they have whooping cough at first until the disease progresses. But this is when they are the most contagious. And the bacteria attaches to the ciliated cells in the respiratory tract, as we can see in the bottom image. It kind of pops between the cilia of the host cells, and it prevents the recruitment of neutrophils and macrophages because of the pertussis toxin inhibiting communication. So white blood cells cannot get to the area of infection easily. And then the symptoms of this disease, it starts with cold-like symptoms, so the mild symptoms, but then it does progress to an intense cough, which includes thick mucus, and then the whoop of the whooping cough is kind of a gaspy cough. And then younger children and babies are more at risk because they have smaller airways, so they're more likely to be more severely affected and not be able to catch their breath. The diagnosis for whooping cough starts with a swab or a suction of the secretions from the person infected, and this would be from the nasopharyngeal area, so the pharynx, or the nose area. And then the media that are used for this, to isolate this microbe, would be a Bordet-Genot agar, which includes blood, potato extract, and glycerol. So all of which would kind of act as nutrients. And then the blood helps because this microbe contains hemolysin. And then the second media that is used is a Regan-Low, and this is a charcoal blood agar. And these can both be seen in the top image there. And then another way to diagnose whooping cough would be with a chest x-ray, and this would be to look for inflammation or excess fluid in the lungs. So in the bottom image there, this was a child who got a chest x-ray, and you can kind of see the cloudy areas in the lungs. Those would be excess fluid. So treatment for whooping cough include antibiotics, which would be azithromycin, erythromycin, or clarithromycin, and these are all macrolide type antibiotics which block ribosome from translating and producing proteins. So they're protein synthesis inhibitors. So that is their specific mechanism is to block the ribosome. And because they're protein synthesis inhibitors, they're most likely inhibiting the pertussis toxin, or the production of it. And then the DTaP vaccine can also be used against Bordetella pertussis and whooping cough, as it was previously for diphtheria. So the final disease I'm going to cover today is Croup, and this is most frequently caused by human parainfluenza virus. This is a single-stranded RNA virus. It is enveloped, and it uses the surface proteins H, N, and F for attachment. So as you can see in the image to the right, the protein, specifically I believe the HN protein, binds to salicylic acid containing receptors on the host cell, so that's how it attaches. And then you can see in the image the rest of the process for replication and release. And then a viral infection from this virus can lead to a secondary bacterial infection due to the body being more immunocompromised, and the resident bacteria that most likely will cause an infection would be Staphylococcus aureus or Streptococcus pneumoniae, and these are both resident in the respiratory tract. So the disease mechanisms, epidemiology, and symptoms for Croup include it spreads through respiratory droplets, so sneezing or coughing. The virus also, similarly to whooping cough, it binds to the ciliated cells in the respiratory tract, and that's where it replicates. But very different from whooping cough, white blood cells will actually infiltrate the infected area, and what that means is there will be a lot that will rush in, almost too much, and this causes inflammation of the airway, as we can see in the x-ray to the right. That is not what a normal airway should look like. It is very inflamed, and this will also cause, as the symptoms listed below state, difficulty breathing, and in small children, cyanosis, which is when they start to turn blue due to lack of oxygen. So young children are more severely affected due to their smaller airway again, and other symptoms include a hoarse cough, a fever, and Croup is actually the name for this disease when it reaches the larynx and trachea area. So before it reaches that far down in the respiratory tract, it is not Croup. Once it gets into the lungs, it's actually pneumonia. And if it's just in the nose, upper throat area, not yet to the larynx and trachea, it would just be a cold-like symptoms. So this disease can be diagnosed through listening to the lungs and characteristics of the cough. And then they would also possibly perform a chest x-ray. So it's kind of similar to the x-ray we saw on the previous slide. And then if they're specifically diagnosing this virus, not just the disease of Croup, they would complete a nasopharyngeal, nose or throat swab of the host. And this is kind of like when you get a COVID test, they stick it up, they stick the cotton swab up in your nose. It's the same thing. And then they would also do, again, similar to if you were to get a COVID test, they would do a PCR test, or PCR to isolate the virus. Or what they would do is, so a second way that they could identify the virus would be doing a fluorescent antibody assay, which is they basically tag the virus with antibodies that are fluorescent. And then the treatment for this disease, there are no antivirals available for this specific virus. So there's no vaccine or anything like that, or any medication that you would get to eliminate the virus. So it's more managing the disease, and this is either using corticosteroids, which suppress the immune system and reduce inflammation. They can use nebulized epinephrine, like in the image of the baby there. Or finally, vancomycin and cefotaxime can be used if there is a secondary bacterial infection. So that is all I have. These are my sources. Thank you for listening.